Good morning, students. Today we are going to discuss about embryogenesis. Embryogenesis means the process of development of embryo and the development of endosperm, together called embryogenesis. After formation of zygote, that is, after fertilization, the male and female get united together and it forms a zygote, and the zygote then develop into embryo inside the ovule and in the ovary. And so after fertilization, ovary is converted into fruit and ovule into seed. So in the seed, the embryo formation occurs and it is followed by the development of endosperm that provide nutrition to the developing embryo. So in the next slide, here you see, today we are going to discuss 3 topics. First, the development of dicot embryo, second, the development of monocot embryo, and then the development of endosperm. Here, first you see the development of dicot embryo. As we know that dicot consists of 2 cotyledons. Dicot seed has 2 cotyledons. And here, the dicot embryo development is quite different from monocot embryo. So here, there is a note also. For after formation of oosporozygote, is the result of fertilization and that develop into embryo. And later on, ultimately the embryo germinate and into a new plantlet or new individual. So after fertilization, the oospore undergoes a period of rest. Zygote is converted into oospore. It is also diploid and it undergoes a period of rest. And this period varies. It is quite different from few hours to several more months, depending upon the type of plants. During the period of rest, the vacuole of the oospore disappears and the cytoplasm gets uniformly distributed. In most of the dicot plant, at first, the oospore elongates and divides by a transverse wall into 2 cells. So first, the diploid zygote or oospore convert into 2 cell structure, and that is called the basal cell or suspensor cell, which toward in the micropyle. So basal cells, another name is suspensor cell, and another is the micropyle, found in the micropylar end, that is called terminal cell or embryo cell. The 2 cells you can say, one is basal cell, another is terminal cell or embryonal cell. Embryonal cell always directed toward the cavity of the embryo sac. Next, here see further division in the terminal cell. Terminal cell then again divided and give rise to the embryo, and the basal cell produces the suspensor. So which one is the play more important function? The terminal cell plays more important role in the development of embryo, from which the embryo is developed. And what is the function of basal cell? It produces the suspensor. So what is the function of suspensor? The terminal cell divides by vertical division, forming a 4-cell linear embryo. In some plant, the basal cell also form the hypocotyl. So when after formation of the embryonal cell, the hypocotyl derived from the basal cell in addition to suspensor. The 4-cell linear embryo now divide transversely. So first the embryonal cell develop into a 4-cell linear structure that is called embryonal cell, and it develop into different, transversely divide into different angle at right angle to the first division and forming ultimately 8-cell structure. So from 4-cell to 8-cell structure, it converted into a 8-cell embryo sac, and it is called 8-cell embryo, and it is called octane stage or proembryo. So what is proembryo? The 8-cell stage of embryo is called proembryo or embryonal mass. Next, the 4-cell to the suspensor. That means we got 8 cells, and out of the 8 cells, 4 cells next to the suspensor are called the hypobasal octant, or it is also called posterior octant. Here you see another 4 cells that constitute or called epibasal octant or anterior octant. So out of the 8 cells, 4 cells are called hypobasal octant, and another 4 cells are called epibasal octane. Now, the epibasal octane give rise to cotyledons and the plumule. So, how in dicot the cotyledon and plumules are arises? Cotyledons are derived from the epibasal octane, and plumule, it is the first embryonal axis from which the stem or branch are developed. Whereas the hypobasal octane give rise to the hypocotyl, except the tip. That means root is derived from the hypobasal octane, but the root tip is not from the hypobasal part. Now, all the octane by forming a wall parallel to the surface, that is called periclinial division, form outer and inner cell. So these 8 cell structure again divided periclinially, and the outer cells subsequently divided by a wall at right angle to the surface. That is, again the outer layer of cells divide anticlinally and forming a peripheral layer of epidermal cell. So the octane cell by periclinial division and anticlinal division develop into a peripheral layer of cell that is called epidermal cell. The epidermal cell again divided into 3 layers. Outer is called epidermis, the dermatogen, the inner cell is called peribleum, and the central cell is called plerum. That means the epidermal cell actually consists of 3 layers of tissue, and outer is called dermatogen or skin, inner is called peribleum, and the central middle portion cell, those are called plerum. The peribleum gives rise to the cortex. As in plant anatomy, we know that plant body is made up of tissue and tissue system. So in main body or main bulk of the embryo, it consists of peribleum that give rise to the cortex, and plerum that give rise to the stele. Stele means xylem and phloem. The basal cell undergoes few anticlinal division, or transverse division, and forming 8 to 10 cell filamentous suspension. So now here we are going to discuss about basal cell from which suspension is derived. The suspensor, how it is formed? First it formed an 8 cell, then 10 cell a filamentous suspensor that pushes the embryo. That means when the suspensor became elongated, it pushes the embryo down to the endosperm. The outermost distal cell, that means it is the lowermost cell, the distal cell of the suspensor situated near the micropyle is much larger, and it is called haustorium. So suspensor has 8 to 10 cell filamentous cell, and the top cell that is the largest, and it is called haustorium. The lowermost cell of the suspensor, which is situated near to the embryonal cell, is called hypophysis. That means suspensor has 2 most important roles. One, at the tip or at the opening of the micropyle, it has the largest cell that is called haustorium, and near the embryonal cell, the lowermost suspensor cell is called hypophysis. Due to the continuous growth of the embryo, it becomes heart-shaped or lobed. That means we know that dicot has 2 cotyledons. That means this heart-shaped or 2 lobed derived from due to the continuous growth of the embryo. And each lobe forms the primordium of a cotyledon. That means it is the beginning of the formation of the 2 cotyledons in the dicot. The mature embryo consists of a short axis and 2 cotyledons. In dicot, we found 2 cotyledons, and at the center, there is a short embryonal axis. In dicot, the plumule occupies the terminal position. So, what is the important characteristic of dicot embryo? The plumule always occurs at the terminal position, and the 2 cotyledons are lateral position. So, here is the diagram. A diagram shows how the zygote is formed after fertilization. It divides transversely into a suspensor cell and an embryonal cell. The suspensor cell by transverse division form 8 to 10 cell suspensor structure, and here is the uppermost cell is the largest. Here you in D2F diagram you see the topmost cell of the suspensor gradually increases in size. It is called haustorium, and the lowermost cell of the suspensor that found near the embryonal cell is called hypophysis. So here the it is the part of suspensor cell that is suspensor that is derived from the basal cell or suspensor cell. Next is the embryonal cell that divide by transverse division and then periclinial division and form 4 cell to 8 cell, then into 16 and 32 cell structure. In this way, it form an embryonal mass of cell, and gradually the outer cell develop into dermatogen, the middle cell into periplane from which the cortex derive, and the central cell that develop pleuron from which the xylem and phloem or vascular bundle arises. So here is the embryonal mass of cell that going to differentiate it into a heart-shaped structure. Here in each diagram, it gradually increases in size from the ground tissue, meristematic tissue, and epidermal tissue, and the vascular tissue, etc., and become it is heart-shaped. And here you see the 2 cotyledons are formed by this heart-shaped structure, and the embryonal axis that is found between the 2 cotyledons, and it is the plumule that found at the apex or terminal position. So this is the diagram of the dicot embryo. Next diagram, it also shows the same structure, and from here you can understand that how the zygote develop into oospore, then into embryonal mass of cells, and into a dicotyledonous embryo. Next is the development of monocot embryo. The development of monocot embryo is similar to the dicot embryo up to the pro-embryo stage, that is the 8 cell stage. In the dicot, it is quite similar, but the monocot embryo is quite different in these 2 groups. In dicot and monocot, the development in later part of the embryo is quite different. In most of the monocotyledons, the oospore divides transversely and the cell divides transversely to form a basal cell, which a middle cell and a terminal cell. So in the monocot, we found 3 cells, that is basal cell, middle cell, and terminal cell. But in dicot, we first found 2 cell structure after formation of zygote when it divides transversely. One is basal cell, other is terminal cell. The large basal cell lies toward the micropyle in case of monocot and directly transform into a large vesicular cell without any division. So here in monocot, the basal cell directly develop into a large vesicular cell. The middle cell then divides repeatedly by a series of periclinial and anticlinial division to produce several superposed tiers of cell, which produce several succession next to the vesicular cell. That means it forms the middle cell play important role in case of monocot embryo. It forms vesicular cell below the vesicular cell, it forms suspensor cell, it forms also radicle and the plumule. So what is radicle? It is the part of the embryonal axis from which root is derived. And plumule, it is the part of the embryonal axis from which stem or shoot apex is derived. The terminal cell gives rise to the single cotyledon. So what is the main function of the terminal cell? It forms the single cotyledon. And what is the function of the middle cell? It forms the suspensor cell, radicle, and plumule. And what is the function of basal cell? It forms only in a single large vesicular cell. So these parts of the monocot embryo is quite different from the dicot. Thus in monocot, the cotyledon is single and it is placed in the terminal position. So what is the difference between dicot and monocot embryo? In dicot embryo, the cotyledons are lateral in position, but in monocot, single cotyledon is always terminal in position. And while the plumule arises as a depression on the lateral side, so that means plumule or embryonal axis lateral in position. So here you see the diagram of monocot embryo development. Here the zygote differentiated into two part. Outer is called suspensor cell and the lower cell is called embryonic cell. Next in diagram, here you see the middle cell. Then from the middle cell, radicle and plumule and suspensor are formed. And terminal cell, the embryo or dermatogens are formed. And here you see the cotyledon is derived from the terminal cell. So this is the diagram, the single cotyledon of the monocot is called scutellum. It is the term only applicable in monocot embryo. And moreover, two terms we always used in case of monocotyledonous embryo. Here, coleoptile and other is coleorhiza. Here, I think that the diagram is cut off. So here, I again move to the previous slide. Coleoptile, C-O-L-E-O-P-T-I-L-E, and coleorhiza, C-O-L-E-O-R-R-H-I-Z. So what is coleoptile? The embryonal axis, that is plumule part or shoot apex, is in monocot embryo always covered by membranous covering. It is called coleoptile, and it is found in monocot embryo only. And what is coleorhiza? It is the membranous covering that cover the radicle or root part of the monocot embryo. So these two terms, I think three terms, scutellum, that is single cotyledonous monocot, one term. Second is coleoptile, that is membranous covering of the shoot apex of monocot embryo, and the coleorhiza, the membranous covering of the radicle of the monocot embryo. These terms are actually used in monocot embryo, not in dicot. So in next slide, here also the development of monocot embryo, how it occurs. Actually, the figure is from our textbook, so you can go through our textbooks also, and the diagram part you can draw from here also. So this is the vesicular cell, these are the suspensor, this is radicle, this part is the plumule, and lower part, that terminal cell that converted into single cotyledon or scutellum. So I think two sections of the embryogenesis, development of dicot embryo and development of monocot embryo, are clear now. Next you see endosperm. So what is endosperm? As you know that in angiosperm, double fertilization or two fertilization occurs. That means first zygote is formed and second endosperm is formed. So zygote and formation and triple fusion together called double fertilization. So in triple fusion, the endosperm is formed. What is the role of endosperm? The endosperm play an important role in the supporting embryonic development by supplying nutrients, protecting the embryo, and controlling embryo growth by acting as a mechanical barrier during seed development and germination. So in case of angiosperm, endosperm plays an important role. How it is derived? It is derived from the triple fusion. And what is its chromosome number? It is always triple. That means its chromosome number is trisome. And it is developed from a pen or primary endosperm nucleus, etc. Here you see the type of endosperm. There are three types of endosperm are actually found in different species of angiospermic plant. First one is the nuclear type. Here, when the pen or primary endosperm nucleus is formed after triple fusion, the nucleus divided repeatedly and a number of nuclei are formed in the endosperm, but there is no wall formation. So these nuclei are floated in the main body of the endosperm. It is called nuclear endosperm. Cellular endosperm, when after the formation of repeated division of the pen or primary endosperm nucleus, each nuclei covered itself by some amount of cytoplasm and cell wall and converted into a cellular structure. It is called cellular endosperm. Next is the halobial type. Here, it is an intermediate between the nuclear and cellular endosperm. That means the primary endosperm nucleus was divided by transverse wall into a two nuclei structure, and in one part, the nuclei divided repeatedly without any cellular development, but in the other part, it divided at many nuclei are formed, and nuclei are separated by the wall and some amount of cytoplasmic matter into cellular structure. So in halobial endosperm, one part is nuclear, another part is halobial. So this is the end of the slide. Now I think you have noted all the important matter of the our today's discussion, that is the embryogenesis. What is the embryogenesis? process of development of embryo endosperm, and the post-fertilization processes are called embryogenesis. So in this discussion, today we have discussed about development of dicot embryo, development of monocot embryo, and development of endosperm. How dicot embryo developed? Dicot embryo developed as from the figure, here you see dicot form a membranous wall around it, undergoes a period of rest, then it is called oospore. The spore, after a period of rest, it divided transversely into two parts. One is suspensor, other is embryonal cell. And the suspensor cell repeatedly divide, forming a linear, elongated 8 to 10 cell structure. And the uppermost larger cell is called haustorium. And the lowermost cell of the suspensor cell is called hypophysis. And the embryonal axis or the embryonal cell, it divides transversely and periclinially and form into 8 cell structure that is called proembryo. The proembryo divided into four tiers of cell. Upper 4 tiers are called epibasal octane and lower 4 tiers are called hypobasal octane. They again divided periclinially and anticlinally, forming 16 cell and then 32 or 36 cell structure. And in this way, it grow regularly, forming a globular structure. And the tissue differentiated into three layers. The outer is called dermatogen, middle is called peribulum, and the central is called pleurem, from which the various tissue systems are derived. That is epidermal tissue system that form the skin, the ground tissue system that form the cortex and ground tissue, and the pleurome that form the xylem and phloem, part of the vascular bundle. So here the hypophysis develop into radicle, and embryonal axis then differentiate into a globular or heart-shaped structure, from which the plumule and the two cotyledons are developed. This heart-shaped structure is the important characteristic of the dicotyledonous embryo. And another important characteristic that the plumule is always terminal in position in dicot embryo, and the two cotyledons are lateral in position. So next, we go to discuss about monocot embryo. Here, what is the difference between dicot and monocot embryo? In early stage, they are quite similar. After formation of dizygote, it forms oospore, then divided transversely into two cell structure, that is embryonal cell and suspensor cell. But after that, the development of monocot embryo is quite different. Here, another middle cell is formed between the suspensor and embryonal cell. So here, a middle cell plays an important role. From middle cell, radicle and plumule is formed, and the suspensor are formed, and the basal cell and the from suspensor cell, one vesicular cell is formed, single vesicular cell is formed. And the embryonal cell, it is differentiated into a single terminal cotyledon that is called scrotalum. So this is the development of dicot and monocot embryo. And then about the endosperm, here it is a nutritive tissue that helps in the growth and development of the embryo in dicot and monocot plant. So endosperm are three different types. It may be nuclear, it may be cellular, or it may be halobial. So this is the end of the slide. I think about embryogenesis, we have already discussed and covered all part. If there is any doubt, you can ask in our Google Classroom. So thank you, students, and thank you very much.